friends, you're welcome back to the channel, and it's beautiful to have you on this bright day. Today we are going to talk about the mechanisms of plasmid transfer. We have a former video where we talked about plasmids, what they are. Please do well to check out that video and understand what plasmids are. We said in that video that plasmids are extra chromosomal DNA. So they are DNA that exist apart from the normal chromosomal DNA of the cell. They exist and they confer particular characteristic on that cell. Now, for today, we are going to focus ourselves on what are the different ways through which plasmids can be transferred. What are the mechanisms for the transfer of plasmids? There are three major mechanisms for that transfer. The first one is conjugation, the second one is transformation, and the third one is transduction. Now, we'll break down these three major processes and understand what they are. Now, in conjugation, conjugation is a process where you have the transfer of genetic materials from one bacteria to another, using a sex pilus, which is sort of a passage from one bacteria to another. Now, it's important to know that plasmids can be transferred through this method if two bacteria begin to engage in a conjugative transfer or a conjugation process where you now have the transfer of DNA and other genetic materials from one of the bacteria to the other. We know that in this process, plasmids can be transferred from one bacteria to another. So this is important for us to know. Now, apart from conjugation, the second process is transformation. And what's transformation? Transformation is a process in which we have the take-up of what we refer to as exogenous or extracellular DNA from the environment into a bacterial cell. So it is very possible that if a bacterial cell stops to show characteristics of life and yet has its DNA within the environment, whether it's in the solution or in the air or wherever it is possible for another cell, a bacterial cell, to take up this DNA. So it's possible for another cell to take up plasmids that are present within the environment. And these plasmids may not necessarily be within a bacteria. They may just be present in solution within the environment, in the air, or wherever they may be. It's possible for a bacteria to take up this DNA. So plasmids can be taken up through that method, and that method is the method we call the method of transformation. You can read up, or we might do a video on, I will do a video on Griffith's experiment to further explain the principle of transformation. Now, after transformation, we also have transduction, which is the third method. And transduction is a method in which we have a process where you have the use of bacteriophages. So bacteriophages can help to transfer plasmids from one bacteria to another, or from even the environment to another. So if a bacteriophage, which is essentially a virus, is able to invade a particular bacteria, it's possible for the bacteriophage to take up some of the plasmids that are within that bacteria. And then if this bacteriophage finds its way into another bacteria, it's possible for the bacteriophage to transfer the plasmids from bacteria A to bacteria B. So this process of the use of bacteriophages to transfer plasmids from one bacteria to the other, is the process of transduction. This is really important for us to know. Now, it's also important to note that in plasmids, there are mechanisms that help to control the number of plasmids that can exist within a bacterial cell. There are normal natural mechanisms that help to control this method, and this is important so that we don't have an overload of plasmids within a particular cell. One of the methods is what we call a copy number control. Copy number control, meaning, and in this method, what occurs is that when one bacteria replicates from being a mother bacteria and goes to having two daughter cells, it copies the exact number of plasmids that are in the mother cell into the two younger or two daughter cells. And this is important. This is a mechanism that controls the number of plasmids that are present within the cells. And this is important to know. There's also what we call partitioning system. So when a bacterial cell is about to divide, as that bacterial cell is being partitioned, we see that the number of plasmids that are on either side of the partition are controlled, are regulated in such a way that one bacteria doesn't carry too many plasmids at the expense of the other bacteria. We also have what we refer to as selection pressure. And here we have the presence of antibiotics and other selective agents that help in the retention of plasmids. and they confer antibiotic resistance on plasmids, and they also control the way plasmids are being, replicated from one cell to another. There are many applications for plasmids, and it's important for us to know some of these applications. One is that plasmids are applied in genetic engineering. So a lot of viral vectors, or a lot of vectors, sorry, not viral vectors, a lot of vectors that are used for the transfer of genetic materials are usually in the form of plasmids. And this is important to know. Plasmids are also used a lot to generate a lot of vaccines. There are different kinds of vaccines, and we'll do videos where we discuss the different kinds of vaccines. So we have different kinds of vaccines, and plasmids are usually used to make some of these different kinds of vaccines. This is important for us to know. Plasmids can also be used in agriculture, where we have the production of GMFs, genetically modified foods, genetically modified organisms, for us to vary the genetics within these plant cells. We use a lot of plasmids as vectors for the new characteristics and traits that we want to bring into these plants. So this is important for us to know. Thank you for listening to our lecture. I hope we understood a lot about plasmids. Like we said before, we'll do many other videos where we try to explain vaccines. We also try to explain other concepts. Please remember to check out for those videos. I hope this helps you in your studies. Remember to like our content, help us so that our channel can grow. Have a wonderful day.